So my name is Andrew Hirzel. My mentor is Nicole Polton, and I'm going to be talking to you about biovolume, cell biovolume today. So why do we care about cell biovolume? Primarily is used to calculate other parameters that we care about in the ocean, so cell biomass, cell growth rate, primary productivity, carbon sequestration, and a variety of other factors. The two that we care most about this are primary productivity, which we use to get oxygen, and that's what we breathe. And that's very important for fisheries because the fish have to eat something, and primary productivity is it's the very base of the food chain. Carbon sequestration, we care about that because of global warming. So the more carbon dioxide that gets into the atmosphere, some of that gets evened out into the ocean, and when it's taken up by plankton, then it is converted from inorganic carbon dioxide into organic molecules, and when those organisms die, they can sink down into the deep ocean, sequestering the carbon dioxide out of the atmosphere. Now, both of those are in bold because neither of them are linear fits. They are in power laws, so small changes in cell biovolume can cause large environmental changes. So one of the instruments I worked with is the Confocal Microscope, and what this does is it takes 3D images of the cell. So shown here is a 3D rendering of one of the pictures, and what you get is when you're taking pictures is each of those 5 images is one of the stacks through the slide. There are about 58 pictures in this particular slide. They range anywhere from 60 to 120 for this organism. And shown in the bottom left is the confocal microscope that is in Dr. Emerson's lab. The second machine that we used for this experiment is the FlowCAM. It is a flow cytometer with a camera attached. So as the cells go through the little blue slide right there, you take a picture of them as they go by, and those all are 2-dimensional rather than 3-dimensional objects. So this creates problems when you look at the pictures, because not only is the orientation going to be different as you go through them, but also you have to extrapolate the 3rd dimension in order to get a volume estimate. And that is what we use algorithms for. Now, the benefit of this is that you can take a lot more pictures much more rapidly. So in the confocal microscope, it takes about 20 minutes to get one image if it's a really big cell. For this, you can get quite a few more, maybe like 1,000 in 20 minutes easily. And it's much cheaper to run. So the first of the algorithms, the 2 algorithms that come built in on the FlowCAM, these are both pre-installed. You can just open up the software and they'll automatically give them out in Visual Spreadsheet. The first one is the AreaBased Diameter. So what it is it takes the area within the pixels, and it converts that area into a circle of the same area, and then it takes that and turns it from the radius of that circle into a sphere, which you get the volume. Equivalent spherical diameter, it takes the diameter across the cell. So you go midway, and even though that isn't the center, it does have a concrete center that it goes and draws this in between. And then it takes the average diameter from that and turns that into a circle with that diameter, then the radius into a sphere again. Now this one does have a bit of problems because like those spines, you'll see how there's a line that goes all the way through it. If it is perfectly along the line of those spines, it will take the entire length of the spines and call that a diameter. So it is inaccurate in that sense. Geometric shape model is another one that people use. So if you look at most plankton, you'll notice that they typically look like shapes. So the Coskin discus up there, it looks like a cylinder. So you can use a simplistic model to make it into a cylinder, guessing at the biovolume if you measure the diameter and the height of the cell as you go. Now, if you have a more complicated plankton, such as the Bioceratium, then you have to use much more shapes. So you end up with 4 shapes for that organism instead of the 1 shape, since it is a more complex molecule. Now, the problem with this is that in order to be increasingly accurate, you have to keep on using more and more shapes. So for example, for the Bioceratium, if you look at the part that's represented by a cylinder right there, and then you compare it over to here, you'll notice that that isn't a perfect cylinder, and that is a problem with these methods, especially when you get into other parts. So like that is not a perfect ellipsoid, this isn't a perfect cone, and so that ends up accumulating errors. And so this is a paper by Hirzel et al., and what it shows is that They did a few different geometric methods, and there's quite a few ways that they've come up over the years to try to make these more accurate. They compared that to confocal measurement on a log scale, and you end up finding out that there's a 30% to 110% overestimation of volume, which is fairly bad. Now, these also take quite a while to actually measure, and for this purpose, we're not doing it for this experiment. However, it is an important method, and it does have significant error in doing so. Now, this method is one of two that we use the image J in order to identify. What this does is it takes the axis, the long axis of the cell, and then it segments that into small segments, and then it spins it around the axis. So you get a bunch of these consecutive cylinders. Then you smooth it out, and it sort of looks like a caterpillar at the end. The distance mapping is used for more complex shells, and of the cells that we looked at in this experiment, only one of them fit the criteria to look at this, and since we had technical area issues, we could not look at all of the cells, so we only ended up looking at one. However, what this does is it counts the pixels in from the perimeter, and with each pixel in, it steps it up one more. So you get something that's sort of pyramid shaped on the cell. And it works very well for complex shapes, so that's what we looked at in the case of the experiment. So these are the 5 cell types that we used, 3 of which are cultures, represented by the CMP numbers, and 2 of which were natural samples that we collected off the coast of Maine. 2 of which are diatoms, the green ones, and the other 3 are dinoflagellates. And so you can see the shapes are vary from cylinders to spheres to more complex shapes. And this is the one that we looked at with the distance mapping method. So we wanted to make sure our confocal microscope was accurate in taking accurate data, because if you'll see the next couple of slides, you'll see that it isn't always the most perfect data. And so we looked at it with calibration beads. You can notice that there is a bead, a 10 micron bead right there, and a bunch of black junk, which is glare created from the microscope from using the microscope. So that does introduce error. But in looking with the actual measurement, you'll see that there is no significant difference. In fact, that's a 1% underestimation if you do the hard math between the means. And so we are fairly confident in our measurements on the confocal microscope. Now, as I mentioned before, orientation in imaging with the FlowCAM is an issue, and what this graph shows, so each of these along the side is the different orientations for that molecule, and here you have a 3D rendering that we did with the confocal, and each of the orientations that we looked at with the FlowCAM. So I've shown you each of them. So what this is this graph standardizes it according to the one I've labeled standard, simply because they all have very different volumes, and so we wanted to look at them all in one graph. And they're all very significant. And this is ABD. And you can see that there is a 50% underestimation for if the orientation is wrong. So we can't actually use the ones here or the ones here in our volume measurements, simply because they are so different than if you get the entire plane of the cell in one picture. So here's all of our data that we've collected in this lab. What you see is each of the pictures is one of the species. So that's Coscinodiscus, Didylum, Alexandrium, Dinophyces, and Serratium. And then each of the bars is a different method. So it goes confocal, ABD, ESD, and then the concentric cylinder. The distance mapping we only did for the Serratium, so that one's only in that slide. You'll notice that for the 4 over here, they're all fairly equal. They're in the same order of magnitude. They are all significantly different with a very, very high p-value. I believe for this one, the p-value that gave me was 0, and since my program gives me back p-values of 10 to the negative 50th, and I've seen that, that means that it's well below 10 to the negative 200 in magnitude. So that is very, very, very significant. But if you look at the actual numbers, they are fairly close together. With the exception of this bar right here, this is a 1000% overestimation in volume for the Serratium, for the one that I showed you, the diameter one, equivalent spherical diameter. And this is because it takes the complex molecule and since it's going to the perimeter, it fills in the blank spaces of the picture and ends up overestimating the volume. So I would not recommend using that algorithm, the equivalent spherical diameter, on the FlowCAM, because it is highly inaccurate and it will throw off your measurements with the other ones. As for the most accurate ones, I would suggest using ABD or the concentric cylinder. Those both seem to be fairly in line with the confocal, though the confocal did have issues, as you can see here. This one's about half the volume of these ones, and we believe that is due to insufficient laser penetration. So here is a simplistic graph. This is just an average of all the different methods that we chose. it isn't actually statistically significant or used as hard numbers. These aren't actually percentages, this is an average, and it's just me combining all the different cell types, so we had 5 different cell types, into one graph so that you can see the general trends of the algorithms. And so since we standardized them all to the confocal, you see that it is 0%, simply because it's standardized to that, so of course it's not going to have any difference from that. Now, if you look at the ABD and the consecutive cylinder one, you'll see that those are both around 12, 36%, and that was on the low end of the geometric method where they use the shapes. But if you look at the ESD, you'll see that with the serratium, it's 213% overestimation on average. Even if you remove that, it's 47%, which goes to show you just how inaccurate that is and how variable that method is. I would recommend not using that method. If you average the ABD and the CEC, you end up with about 20% overestimation, just looking at this graph, and I've crunched numbers. If you remove the error that we saw in the Koskinen discus, then ABD gets even closer. That only ends up being about a 2% difference from confocal. So that is highly comparable to the confocal, and I would recommend using that. That one's really easy to calculate if you're using the FlowCAM and using the visual spreadsheet. The consecutive cylinder one, that is a bit harder to calculate, simply because you do have to use ImageJ and a plugin. However, that one is also fairly easy to calculate, and I would suggest that you use both of them combined in order to get the most accurate, so that if one of the algorithms goes crazy and ends up with a far different method, it gives you far different exaggeration of the volume, the correct volume, it will reduce your error. So I would like to acknowledge the FCM lab, so Nicole, Laura, and Ben, as well as the NSF and David.
There's time for one question.
Do you think that the fact that the calibration beads are all spherical, and some of the challenges are with the non-spherical shapes, affect like how well these algorithms work? It could be improved if we had little like serratium-shaped calibration beads.
The Alexandria, the one that I showed you before, that one is approximately spherical in shape, and that did show a wide variation in it between the ABD, between the consecutive cylinder one, and between the various methods. So
Can everyone hear me? Good. Okay. So, hi, my name is Deanna Holman. I come from Husson University, and I've been working with Eric B. Kraft, Maria Pakiadaki, and Ramona Stepanauskas on the characterization of a microbial dark matter phylum, Woosarchaeota. Okay. I'd like to start my story with this tree of life here that represents all of the known genetic diversity on Earth. And the vast majority of that diversity is prokaryotic in nature. And this tree does a good job of representing, here are eukaryotes, which are plants and animals and fungi, and all of the rest of these are prokaryotes, which are simple cells without a nucleus. And despite their abundance and diversity, we know next to nothing about them. In fact, many of these lineages aren't even cultivated. And because we know they're there, but we don't know anything about them, we refer to them as microbial dark matter. And recent efforts have been made to characterize this microbial dark matter because it's important evolutionarily and ecologically to understand these organisms. And just within the last century, we found an entire domain of organisms that's mostly microbial dark matter, the archaea. So this graph does a good job of representing here are eukaryotes, again, where we are. And all of this is eukarya. And it's been hypothesized that archaea are actually our closest prokaryotic relatives, which is pretty cool. So my project this summer has been working with this larger project to characterize a specific archaea phylum, Vosarchaeota, which is here. Prokaryotes are major drivers of geochemical cycling, and they're important to study for that reason. And archaea, not to be left out, are nitrogen cyclers, nitrogen cyclers, or ammonia oxidizers, an example of which is Thamarchaeota, and methane cyclers is methanogens, an example of which is the superphylum Vosarchaeota, or Euryarchaeota. Vosarchaeota is a recently discovered phylum. It was characterized by Castell et al. In early 2015, and this is just a phylogenetic tree that they drew. Vosarchaeota is here, and it just represents their evolutionary history of Vosarchaeota, or what they think that it is. And they named Vosarchaeota after Carl Woese, who was a major contributor to the classification of archaea. And thus far, Vosarchaeota have been found to have small genomes and cell sizes and to be found in a variety of environments. Castell originally found them in Rifle groundwater, which is a subsurface aquifer in western US. And since then, they found them in high altitude lakes and Arctic permafrost. And we found them for the very first time in marine environments. Current lineages of Vosarchaeota are represented by genomes from metagenomes. Genomes from metagenomes are composite genomes that are representative of all of the genetic potential within a population. And this comes with several issues. Anytime that you combine several organisms into one picture, you're going to lose the natural diversity and divergence of those individuals. And so their phylogenies and metabolic capabilities that come from metagenomes might not accurately portray individuals. Furthermore, metagenomes often use controversial ribosomal proteins for their phylogenies. And so it's important to note here that Castell et al. Characterized one of their organisms, AR20, and this is a cell diagram that they drew. And they found several missing pathways, such as glycolysis, TCA. They didn't find any acid metabolism. But what they did find was a putative hypothetical morin protein, which is here. And morin is a toxin that's pathogenic to bacteria. And you can see it down here kind of attacking the gram-positive bacterium. Single-cell genomics helps to solve the problems of metagenomics by ensuring that the genetic material we're using is coming from a single cell. And we do this by collecting from the environment and then running the sample through the flow cytometer, which essentially funnels the cells into these single cellular droplets. And a laser analyzes that and determines whether there's 2 cells or 1 cell in each droplet. And if it's 2, it tosses it. And if it's 1, it sorts it into individual wells. From there, the individual cells are lysed and then amplified with a non-PCR-based whole genome amplification. And this leads to the ability to construct phylogenies, reconstruct genomes, archive DNA, and all of this eventually leads to the characterization of the physiology, ecology, evolution, and biotechnology of our organisms. Single-cell genomics allows us to have a decreased risk for contamination by other organisms in the environment, and it also is better at analyzing the individual metabolic capabilities and evolution of singular organisms. At the beginning of the summer, I was given 24 archaeal genomes that we thought might be Vosarchaeota, and they were collected from various sites around the world. Here we have some gold mines some subsurface aquifers. This is a hydrothermal vent. There's also a site over here in this oceanic gyre in this one. And we kind of designed a project around characterizing the ecology and evolution of this novel Wolfsarchaeota phylum. And we did this with single-cell genomics for collection and isolation and amplification. And then from there, we used various programs to analyze these genomes. And the first thing we did was construct a 16S maximum likelihood tree, which is depicted here. And color blocks represent individual phyla. And I don't know if you can see it because the slide's a little, but hexagons are my genomes and circles are genomes from the literature, Castell et al.'s genomes. And we found 14 of the 24 genomes were Wolfsarchaeota, which is here. And we discovered 2 new candidate phyla, and we found 1 Pacearchaeota and 3 Microarchaeota, which are possibly the same species up here. And we performed a couple analyses on the Wolfsarchaeota, the average nucleotide identity analysis and the 16S rRNA identity analysis, which essentially measures the similarity between the organisms. And we found that, in fact, the Wolfsarchaeota are not very related. They, in fact, for quite a few of them, they couldn't even be calculated. They clearly are more related to each other than they are to other organisms, but they're not very related. And so based on these findings, we hypothesized that Wolfsarchaeota is actually representative of multiple phyla, that each of these clades are their own phylum, and that Wolfsarchaeota is a superphylum. So the next thing we did was to analyze the metabolic capabilities of our organisms, and of specifically Wolfsarchaeota. And this is a composite cell diagram that I drew of all of Wolfsarchaeota. And dark blue are pathways we found, light blue are pathways that we didn't find. And so the first thing was we determined that Wolfsarchaeota have small genomes, like has been found in the literature. But up to half of these genomes are composed of hypothetical proteins. Essentially, we don't know their function. And these proteins have putative, toxic, and adhesive domains, which is pretty cool. We found a murine protein, much like Castell. And again, that is pathogenic to bacteria. We also found an RTX protein, again, pathogenic to bacteria, and a collagenase-like protein, which breaks down collagen. Something we didn't find in any of the genomes was lipid synthesis. And clearly, lipid synthesis is very important because your entire cell wall is composed of lipids. This could be because of the nature of single-cell genomics, we aren't working with full genomes, the partial genomes, so we may have just missed that part of the genome. It may be that they're using hypothetical proteins to produce their lipids, and we just aren't capable of detecting that. It also may be that they're just missing the pathway and that they're getting their lipids from environmental sources, which would be pretty cool. We also detected flagella, which means that our Wolfsarchaeota are potentially mobile. So based on all of these findings together, we hypothesized that Wolfsarchaeota is breaking down organic matter. It's getting metabolic needs from the environment. And additionally, that they may be pathogenic to bacteria, which would make them the very first example of pathogenic archaea. And I would like to thank the Bigelow REU program, my mentors, and our collaborators, and of course, David Fields. What? Oh, any questions? Any questions at all? No? Yes.
So they rely on a gram-positive bacterial community. Do you find any evidence of co-distribution with gram-positive bacteria in Wolfsarchaeota?
I didn't look at that. I think that maybe that would be something for future directions, but my job this summer was just to kind of take a brief glance at it, so it's not something I looked at, unfortunately. Any other questions? Yes.
Yeah, I had a question. You showed that they were actually pretty distantly related.
Yes.
Really interesting. But did you find those same murine proteins in all of them?
No. No, we found it in one of the SAGs. It was one from Oasis Valley 2, which is a subsurface aquifer, I think. But I mean, it was similar to Castell in that they also found a moraine protein, so we thought it was important to note. Anyone else? Yes, David.
So you get a lot of viruses that attack them, and do viruses also attack the archaea?
I would assume so.
And so is it possible that some of the genes that you find are not characteristic of the archaea itself, but could be just viral genes?
I don't know the answer to that question. Yes, Maria?
I